Well, thank you everybody for coming. I know it's a small group today, but hopefully that'll allow us all to interact much better. It is final season, so I suspect there's a lot of competing activities at this point. But yeah, thanks for joining us. George is presenting today. George is a PhD candidate in Dr. Fitak's lab in the University of Central Florida. So if you recall from CCD seminar series not too long ago. Prior to pursuing his graduate studies, George completed his undergraduate degree at UC Davis, and he gained several years of industry experience as well, working for Bayer Crop Science. George is broadly interested in understanding how human activities affect the evolution and distribution of wildlife, especially managed species. His current research focuses on studying the adaptive potential of Florida panthers, as we've heard already, and white-tailed deer under future climate and land use scenarios. And he is also evaluating the impact of urbanization and evolutionary divergence in Florida key deer. So if you have any questions about that, he's the person to ask. And yeah, thanks so much for joining us. And yeah, join me in welcoming George.
Right? Yeah. Yeah, thank you. Thanks for the invite. I'm glad I was able to come in person, and I originally was going to do this virtually, but then ended up planning a trip to visit my family for like the holidays, and things just lined up. I was able to get travel funding to come here because it was part of a talk, and so I got my trip to visit my family paid for by the university. But it was a win-win either way. Today I'm going to talk a little bit about how to estimate and visualize population genetic structure using a method that I think is really useful, especially for some preliminary analyses you might be curious about for your data. Share your slides. Oh yeah, sorry. I didn't realize I'm not sharing yet. Okay. All right. And so at first I wanted to give a little bit of a background about people for people that might not be familiar with landscape genomics. But in a nutshell, it's a study of the interaction between landscapes and genetic variation. This term landscape genetics and landscape genomics has been used for some time, but really the person who, or the people that coined it were Stephanie Manel and her group back in 2003. Really trying to bridge some of these aspects about trying to understand how fragmentation or landscape like patches affect biodiversity, and using that knowledge in combination with some of the genetic data that people had been generating throughout the years as technology advanced. In 2013, this idea of landscape genetics, where previously you were focusing at just one or a few loci, with the advent and emergence of new technologies, people started looking at more and more loci, where they're looking at thousands to tens of thousands to hundreds of thousands SNPs. And then landscape genomics was really born from this development of that technology. But Stephanie Manel and her group, based out of France, are really, really influential in this entire landscape genomics area. And they're really behind a lot of the development of the software that we'll be talking about today, and some of these ideas about how to analyze population genetics data. All right, so why include spatial data? One of the reasons is that a lot of genetic, or most genetic data, has some sort of spatial context to it. Like I mentioned before, how landscapes are arranged, how fragmentation can occur due to things like urbanization, or how habitat loss might occur due to the expanse of man-made structures, all influence what type of biodiversity might actually exist in those patches. And that influences the size of the population, which thereby influences population genetics dynamics. And so the populations in general are extensions of that environment as well, where environmental characteristics, be that man-made structures or things like climatic factors like rainfall, precipitation, temperature, all of those can affect how a species interacts with the landscapes and affect the outcome of genetic variation that we see over time. And so understanding the spatial context is really important when trying to understand the evolution history that might be shaping the trajectory of species. And so when thinking about landscape genomics or landscape genetics data, there's really three types of data that we work with. One is geographic data. So this is data that looks like a map, is typically some sort of landscape characteristics, be that geology or things like climatic factors that are displayed on a map. There's genomic data, which includes like your DNA sequence data or your SNP data from either RAD-seq or genome-wide approaches or even microsatellites. And then there's environmental data, which if you're doing if you're planning to do scans for selection or adaptation, you can include things like environmental data, like climate, like temperature and precipitation, to then see how those might be assessed with certain outlier loci. So for geographic data, most of what we work with is based out of using geographic information systems. How many people here actually use things like GIS in their research? Cool, so there's like half-ish of the people. So GIS is really, really important when working with landscape genetics and landscape genomics data, because it's just so easy to like manage that type of data, especially when you're like working at multiple spatial scales. And then you can build that into a pipeline where you can analyze your data in your series of scripts when you include genomic data, and then you try to also include things like the geospatial data. GIS helps you analyze these spatial relationships that might occur in genetic variation. So you can then assess how is genetic variation in one area of a island, for example, how does that compare to the genetic variation in either another area of the island or in a different island? And so that can help you assess what's going on with that genetic variation, also develop hypotheses that you might use to try to explain some of the evolutionary processes behind the genetic variation that you see. Geospatial data often comes in the forms of what's common is telemetry or tracking data. So this is P22. Have people heard of P22? I'm pretty sure California people have definitely, at least most people. Okay, so P22 was he was a mountain lion. So background: Puma concolor is a species that's behind mountain lions, cougars, Florida panthers, etc. There's I think over 80 common names for Puma concolor. But he was a puma that was isolated in Griffith Park in LA, and so he lived in Griffith Park I think for about a decade, from like 2010 or so up to 2022. And so he got really famous locally. People like took pictures of him like wandering around Griffith Park and around their neighborhoods. And over time, people like ended up really, really appreciating him being there, and he developed a big following. What ended up happening was, unfortunately, he was over time the victim of a few vehicle collisions and diseases. And I think a vehicle collision was ultimately what caused his death in 2022. And the California Wildlife Agency decided to put him down. But he's a panther that was really, really publicly observed for quite a long time in Griffith Park. And in order to get to Griffith Park, he had to cross two major freeways in LA, which was kind of like a big deal, because it's like, I think to this day, people are still kind of trying to find or really explain or have hypotheses about how he could have got to Griffith Park. But he was really, really famous. There was a whole celebration of life after his death in 2022 at the Hollywood Bowl, where they had like celebrities. Who's the guy from The Office? The Dwight in The Office. Who's the actor that plays him? Yeah. So he was at the Hollywood Bowl, like singing songs about P22 and things, and it was actually really cool. But so P22 is one of those famous pumas, but he was tracked using these telemetry collars. And so a lot of these with Florida Panther data, for example, back where I'm from at UCF, with the Florida Fish and Wildlife Commission, they use a lot of these telemetry collars in order to track the Florida panthers. So when they go out and sample species, they can go out and get samples of blood or tissue, and then also get the geospatial coordinates associated with those. So then you have potentially genetic data, or you can extract DNA to use genetic data in combination with that geospatial data. Traps are also really commonly used. You can have traps set up in a landscape. One of my colleagues, another grad student at UCF, she has a study on pygmy rattlesnakes. And so she has traps set up, and she can go out and collect like venom or other things from the snakes, and then have the data for those geospatial coordinates where they are found. But that's geographic data in general. So one of our three types of data, and our second type of data is genomic data. So genomic data, especially with landscape genomics or landscape genetic studies, primarily comes in the form of SNP data. So early on, in the early thousands, it was more so microsatellites that were being used for a lot of these like early landscape genetic studies. But as technology has progressed and there's easier access to whole genome sequencing, you're starting to see a move towards whole genome sequencing data in these landscape genomic studies. This started, I would say, about a decade ago, really with a lot of livestock species, and that's just a matter of funding. Livestock species are funded by agricultural people interested in agricultural development and industry. And so from there, it started to expand into a few more non-model species. So it's kind of nice to see whole genome sequencing data included in these landscape genomics approaches. And one of my projects, or a couple of my dissertation projects, are using whole genome data of Florida panthers and white-tailed deer with using that whole genome data in order to design these landscape genetics type or landscape genomics type of studies. But that being said, RADseq is really commonly used. RADseq is probably the most commonly used method. If you read any paper on landscape genomics or genetics, you'll see RADseq. You identify however many thousand SNPs, and then you run that through a pipeline. A lot of different ways that genomic data can be incorporated or generated to use for these types of studies. So the third type of data is environmental data. So most of these are often from global environmental data sets. So does anyone here use things like World Climb or NASA Landsat data for any of their stuff? Cool. Yeah, so these are two popular databases. World Climb has a lot of bioclimatic variables, so things like precipitation, temperature, lowest winter temperature, highest summer temperature data, all down to, I think, a 1 kilometer square resolution, but up to other scale resolutions as well. And what's nice about databases like World Climate Data or the World Climate database is you have future projected data up to 2100. So then you can design predictions or develop predictions about how distributions or genetic adaptations might change over time and what types of populations might be at risk. NASA Landsat data includes a lot of things like vegetation data, where you can then assess how much of canopy cover is covering a landscape, and that does that affect the distribution of what you see in terms of your species or your genetic variation. There's regional GIS databases, so a lot of this is basically just Googling and trying to find what's available in your area. A lot of state agencies often have a database with GIS data that's available. These are usually down to lower scale resolutions and include things like local hydrology, soil types, and topography of the landscape that you that are using. But again, this is another type of data set that you include usually if you're doing some sort of analyses where you're trying to assess some sort of adaptive evolution question. All right, so those three data sets. So we have our geographic data, our genomic data, and our environmental data. So our geographic data often looks like this. We have some sort of animal ID and some latitude and longitude coordinates associated with that. So that's based off of things like our telemetry data that we gather. We have our genomic data where we have SNPs that are in some format, but generally you have a column representing each SNP and a row representing each individual. And then you have data for those individuals across however many SNPs you're trying to analyze. Then we have our environmental data that I mentioned. Here you have an example of elevation, precipitation, temperature, and you have that again for each of the locations that your individuals were found. But really, really common to see your data format look like this in any of these types of studies. All right, so how do we use this data to understand evolutionary processes? One way that's really popular is assessing adaptive evolution. Where you try to perform something like a gene environmental association, where you're trying to find environmental variables associated with certain SNPs. And that tells you that something about, hey, those SNPs that we found might be important for adaptation to something like temperature that was really associated with it. This data can also be used to assess non-adaptive evolution. So looking at spatial genetic structure across a landscape and trying to assess how that relationship might be distributed between or amongst individuals and trying to get an idea about why that relationship might exist there and generating the hypotheses to do that. But usually when you're designing any of these types of studies, one of the first things you're going to do is assess this neutral evolution and try to assess the general structure of your populations that are there before moving on to your adaptive approaches, where you're then looking for those outlier losses or you're using things like GEAs. Yeah, so today I'm going to focus with the TESS example, mostly on assessing this non-adaptive or neutral evolution by looking at how genetic structure is occurring on a landscape. All right, so to analyze genetic structure, one thing that's commonly used is something called ancestry coefficients. And so this is just the proportion of an individual's genome that come from multiple ancestral gene pools. And so for an example, this is a study by Chan et al. Where they have frogs in the Philippines, and the frogs were assigned to one of five populations based on their SNP data. And so across the islands, what we see is that the pie charts that we see here are representing individual frogs and are color coded by their relatedness to some sort of ancestral population. So we have certain frogs in certain regions that are related to this purple cluster, have ancestry from that purple ancestral group. We have other frogs that are from another island that are from a blue group, and then other frogs up north that are descended from this ancestral population represented by that orange group. And so a lot of these population structure types analyses look a lot of like these pie charts where you have that genetic variation represented in visually that way. One important thing to really note about genetic structure in these types of analyses is that the structure can mimic selection. So often if you see a population, let's say, it's a good example, let's see blue's a little spread out. Let's see this, say this purple one. So we can see this purple population, and there might be some SNP that's only associated with that purple population. Well, that SNP could have gotten there either through things like genetic drift or through things like local adaptation to the environment on those islands. So teasing that apart can be tricky, and it's important to just acknowledge that when you're doing these types of structure analyses. So here's another example. We can have a population that has about even amounts of a variant coding for some sort of blue coat color in a deer, and another variant coding for for a yellow color. And over time, we see that this yellow color increases in frequency in the population. That can occur, again, through either genetic drift or through adaptation if that yellow coat color contributes to fitness in some way. There's two methods that are really popularly, or there's two methods that are really popularly used with analyzing the spatial genetic structure. The first method, probably people here, especially if you're doing some sort of landscape genetics study, or if you've done some sort of landscape genetic study in the past, have heard of structure. How many people have heard of structure, at least read about it in like papers? Cool, so that's most people. Wait, Monica, did you raise your hand or no? Okay, no worries. You're like playing with your hand, so I wasn't sure. But structure was originally developed in 2000, and it's really, really powerful because it uses this Bayesian methodology to simulate ancestry coefficients and determine the ancestry coefficients that are most likely to represent your observed data. It's considered a stochastic algorithm, things like structure. And so it can be really, really slow because it's run using these MC Markov Monte Carlo chains if you're using a lot of SNPs. And so one of the reasons some spin-offs to structure developed, like admixture, is because admixture is not stochastic like structure, it's a maximum likelihood based estimation method. So admixture is a little bit faster than structure, which increases like people's ability to actually use it with larger whole genome-wide data sets. TESS, on the other hand, the original TESS was developed by Chen et al. In 2007. It's considered a model-free, and that's more of semantics. It's model-free in that it's not changing its evaluation of estimating ancestry coefficients based on its data. It's basically condensed down a regression that compares multiple possible ancestry groups to the actual observed genetic variation that you see in your data. TESS accounts for geographic distance, which is one of the powerful things about it, whereas things like structure do not account for that geographic distance. So structure, you might have noticed, especially if you've read that literature, that it's not necessarily used in landscape genetics. People can use it for a lot of other things as well. Whereas TESS, you need to have that spatially explicit data in order to actually use it. What's useful about that is you have geographic distances that you can then incorporate into those analyses to interpolate ancestry coefficients and relatedness across a map. TESS3 was then developed in 2016 as a R package called TESS3R. And TESS3 basically is based off a lot of the similar methodology off of TESS, but is now more optimized to run some of these regression type analyses and maximum reduction of mean squared errors a lot more quickly than TESS had originally. All right, so there's three types of spatial, there's a couple ways to represent spatial genetic data, but here's three types of ways that Melanie Nakawakawa et al. 2021 had chosen to represent it. So Lakawa 2021 was looking at genetic structure in mule deer in Wyoming. And so there was individual coordinates for mule deer that were spread across Wyoming. And then she analyzed genetic variation using structure. So what she saw is that there were most likely three ancestral groups that represented this upper level of hierarchy that are represented by these green, orange, and purple groups here. And so again, this is really commonly used in structure, these types of pie charts across the landscape. And you often see something like these bar plots that have individuals represented by each individual column, and you have some sort of admixture or assignment of ancestry for those individuals. And so what she saw here was essentially that there was a southern group, a northern group, and a western group, which you can see on the map itself has some sort of spatial representation. TESS3, on the other hand, is run using this spatial data in combination with your SNP data. And so that data can then be interpolated on a map. So you can have gradients that represent the probability of finding individuals with ancestry from those ancestral groups, color-coded by these like darker colors, lighter colors in these shades of green, orange. This is from supplementary material, and it's not like a huge deal, but like I wish these colors were switched. Yeah, yeah. I'm glad a couple of people like realized that, but yeah, not a huge deal. But the main important part is that this is spatial genetic relatedness probability, or the probability of a certain ancestral group represented on a map, which is nice to see that whole distribution. This is mule deer, yeah. Otocoileus hemiomus. All right, so now we'll switch to the demo. So this is a really, really quick demo. This is the only math I'm gonna show. So TESS is available as this package I mentioned, TESS3R, and it estimates ancestry coefficients, which are represented by this Q variable, using a least squares minimization algorithm. So what it's trying to do is look at your observed data and then factor out your matrix of observed data, which is represented by this X, into two matrices that are represented by potential ancestry coefficients, Q, and your potential genotypic frequencies in ancestral populations, so G. And so what you have really is X equals QG in a nutshell, and you have X as your observed data, and you want to minimize the error between your observed data and the QG possibilities that TESS generates. And so that's done using like a matrix factorization, which is a little bit beyond like my knowledge of math. But because of that, it's not model-based or not simulation-based like an MC-MC chain like you would see in structure. All right. So now I think this is a good time to, actually we'll talk about this a little bit first. So we're using the built-in example of Arabidopsis thaliana data that's available in TESS3. And so there's two types of input data that TESS uses primarily for this. One is genomic data, which is in the form of a genotype matrix, and one is geographic data with that latitude and longitude, like I mentioned a little bit earlier. And so now, for people that have downloaded the packages, if you can switch over to the code.
So I personally had trouble downloading TESS.
Okay, no worries.
Yeah, and I know like people like barely have laptops and don't have laptops, so maybe we can just go along and like we're recording it and putting everything online.
Yeah, that works. All right. So first thing is going to be just loading those packages. So calling library or calling tester maps, these other map visualization software or libraries. And then next is selecting the data. So now we're just grabbing the data from TESS, which is the example data.at. AT stands for that Arabidopsis thaliana. And then for that data, if you want to take a look here at the actual data, see what it looks like, we can open up the description of the data. And so we see all that data.at is a list of two types of data sets. One is this representation of SNP data. So this is our genotype matrix. And this looks, oh, that's not, I want to see it. Let's first assign the genotype matrix here, make things a little bit easier. All right, so one is the genotype matrix data. So that's something that looks like this. So in this genotype matrix, what we have is our individual columns that represent SNPs. So Arabidopsis thaliana is a haploid organism. So we have one SNP per locus here. And then we have our individuals, which are 170 individuals represented by our row. But this is actually a data set for more than 26,000 SNPs. We also have our coordinate data. So we've loaded this into that variable called coordinates. So if we take a look at that, we have our coordinates representing our longitude and latitude in each column, and then we have rows representing individuals. So we have 170 rows, just like we had 170 rows with the genotype data. All right, so one of the first things that I like to do is just plot that data on a map. So using plot, we can plot our coordinates on a map. Not a lot to see there, just lots of dots. So add a little bit of context by providing a map of Europe. And so now we actually see that these dots represent individuals that are located across Europe. So all 170 individuals are represented by these dots. So we have a little bit more context about what we're working with here. Let me check what questions I had. Oh yeah, so I just went over that. Cool. All right. So right now, I'm just going to start running this line before I talk about it because it can take a little bit of time. So I'm going to run the test itself, test3, which is represented by this function test3. And so it's currently running. It'll probably take about 8 minutes or so. And so test3 has a few functions and arguments, but it's actually really simple, a simple function to run. So we have X that's represented as a genotype matrix. So that's the data that I showed with those zeros and ones. We have our coordinate data, which is our latitude and longitude for each of our 170 individuals. And then we have our number of possible ancestral populations we'd like to compare. So this can range anywhere from 1 population to 10 population, which is really common. But you can add more if you want to. Often there's some sort of decrease in really like the information that you're getting if you run the program longer with more K groups. So like in structure and admixture, a good ballpark rule is just to limit yourself to 1 to 10 populations, and then go from there if you think there's if there's more evidence of like lower hierarchical structure. Method is just referring to that least squares algorithm that I mentioned with that equation a little bit earlier on. And so each of those variables is estimated using some sort of algorithm, but this is basically that standard algorithm that is mentioned in the K 2016 paper. Ploidy, we're working with haploid individuals, so we have just ploidy equals 1. This isn't super necessary. It depends if you're working on something like a cluster where you want to like determine the number of cores that you're using for analysis. But for now, you can ignore that with your computer. It'll default to 1. All right, so choosing the optimal number of K is tricky. So K, like similar to structure, K is determined using this cross-validation criterion. So with test3, it's used using cross entropy, which for test3 partitions that genotype matrix into training data and test sets, and then compares your predicted frequencies based on its optimal values of your Q or your ancestry coefficients with the actual observed data in your genotypic frequencies in that observed data, and then generates how accurate its assessment of how many ancestral populations there are. Smaller values generally imply better estimates in tests. So here we have a cross-validation score output, which usually looks graphically like this. We have a validation score on our y-axis, our number of ancestral populations on our x-axis. So what you often see is a pattern that looks like this. And so here we have 1 ancestral population. I forgot to put the actual like x values. But this is 1 ancestral population, 2, 3, 4, 5, 6, 7, 8. And so as you move down this graph, you notice that this starts to plateau over time. And so one ballpark, which I'm a little bit more on the fence about this, because if you run structure, you know there's things like the Abano method, which are a little bit more robust and quantitative about assigning these population groups. With TESS, that doesn't exist in that way. And so TESS is a little bit more qualitative in terms of we're trying to find the point at which this data or these dots of ancestral populations plateau. And so in this example, that would be somewhere around 5 or 6. I do have to say that the K et al. In that paper, that 26 paper, do explicitly mention that just because you choose a certain number of ancestral populations doesn't mean that there lacks any more structure that exists in your population. And so they're more worried about people being open and honest about assessing different levels of hierarchy and then just admitting that, okay, there could be potentially more levels of hierarchy if we stick to just 3 or 5. Yeah, there can be many that are at smaller spatial resolutions. But as long as you're open about that and don't make any assessments like, oh, yep, optimal 5 looks right, we're going to stick with that, and aren't like super glued into that, then you're on the right track. So it is a little subjective, but it is based on this quantitative metric of this cross-entropy, cross-validation score. And oh yeah. Yeah, so that'll depend on your organism, then like the number of SNPs that you have and like your ploidy, like with like haplo or diploid with diploid individuals, I noticed that there tends to be more evidence of like not lower level, more hierarchical structure. So you can have a lot of populations that are like across some sort of topographically complex area of a map, just as much as you could have like your large populations separated by big geographic barriers. All right. So before moving there, yeah, so this is run. So right now, TESS has been running for, I'd say probably like 4 or 5 minutes. And so what it's here, it's basically computing those matrices that I mentioned a little bit earlier. So let's go back here. So it's computing those Q and G matrices for each number of K populations. So we have, for this example, we're looking at 1 to 7. And so each time it does that, it generates this little like feedback that's like, okay, I've generated this matrix for your first group. You're generated this matrix for your second group, your third group, and so on. So right now we're at about 6 groups. And so the more groups that you run, the longer it takes, because each increased number of K, it takes longer. But I think right now is a good time to just pause while that finishes up for questions. So up to this point, what questions do you have about running SNP either through R or assessing the amount of optimal number of K populations? Yeah.
Questions. First, you said that it takes in, it doesn't take in biallelic data.
No, so it does take biallelic data.
But you're running it as non-biallelic data right now.
Yeah, and that's based on, because this is from Arabidopsis, which is this is Arabidopsis thaliana. Oops. Right, I need.
I don't know why they said, you said that it's from the beaver, right?
Yeah.
So I wonder why they say it's. I was wondering. It's a plant. So I wonder why they said that. But I'm kind of curious how it handles haploid versus diploid organisms in terms of data.
Okay. So you're talking about the actual like let me pull that up actually here. The actual genotype matrix. Yeah, so have you worked with genotype matrices like this before? Okay, cool. Yeah, so we have our haploid SNPs represented by like this. So here's SNP1, for example. We have this like B1. If it's working with diploid data, then you would have a column for each of your alleles. So you would have like SNP 1.1 and SNP 1.2. And so you would have twice as many. Instead of 25,000 rows for a haploid organism, you would have 50,000 or so for diploid, or sorry, columns. Yeah, so so there's ways to convert between data formats. Like I think one of the examples I showed in like my slides was like a simple format like this. And so you can convert something like this into your genotype matrix with, yeah. Yeah, I think I know there's packages out there that exist that allow you to convert between them, but I think TESS has some conversion between types of or data formats as well. But yeah, so it would look really similar for a diploid organism as well. All right. Other questions? Are people getting it to run smoothly on their computer? Is it still running for you all? Oh, nice. Yeah, because mine just. You guys have better computers than I do.
So you just have the genotype information, but you don't have the position, so it can be like empty or something like that.
Do you mean the coordinates?
Yeah, like for the genotype data, like you don't know like if they are on the same chromosome or something else.
Oh yeah. Yeah. So we
I'll make sure you need to encrypt that. So I just.
Yeah, so for TESS, the actual like TESS function, you don't need the locations of your loci.
But yeah, so do you know like what happened if they're all close together?
I'll talk a little bit about that like in the final slides. But there are ways that you can visualize and see if your SNPs are like grouping close together in certain areas or not. Let's see, so mine is done. Yeah.
I see maybe there's a different method, like kind of like a box where it doesn't really plateau, but it's like a little curve.
Yeah.
Like, I don't really know why that happens.
No, I've seen that.
Any insight?
No, I've seen that happen too. Like I've like messed around with data and done like 1 to 30K populations because that makes no sense. But yeah, so you like sometimes see that curve, and I think it's just a matter of like over parameterizing your models.
Yeah.
And so like you end up getting models that are giving you less information as you start including more and more data, because it's just like hard for it to like assess like the not the assignment of those individuals into those populations.
I see why they're running it as a haploid. Because it is a diploid, but they have a high level of inbreeding, so they don't, they assume that at most steps it will be homozygous, I think.
Yeah, I think
Interesting,
also
okay.
the fact that they're using some fertilizer. So it's literally-
Then it's,
So it's like highly
they're
inbred.
not crossing.
Okay.
Like they cannot cross, but.
But-
what's under-
but then-
So. But that's a big assumption.
looking at this map,
Yeah.
I have
That's a
a
big
very,
assumption that
it's a very big assumption.
Yeah,
That's-
cool. Yeah, I didn't know that.
It can be both, but the one that they're using is like.
Yeah, but the only way to ensure that it's solving is,
If you've literally been selling it.
yeah, but like if it's, well, relations, I don't know.
Maybe,
Yeah, they basically just cut their SNP data set in half.
I think that's why they did it.
It doesn't
Yeah.
validate the SNP data set. It's just,
That's
like, they got rid of the information.
Yeah, it does make it like quick to run, especially for like a
Yeah.
demo. Yeah, I don't know. I'm not a plant person at all, so that's good to know. Plants are confusing to me. The multiple chromosomes that you can have is scary. Like I said, I don't know of anyone that works on ferns. That would be intense. Because there's like a species of ferns that has like 1,200 copies of each chromosome, right? Yeah, that's what it was. Yeah, anyways. Any other questions before I move on? Cool. So we just finished running tests, and now let's take a look at those cross-validation plots like I mentioned. And so we're seeing something that looks not entirely obvious that there's a plateau. I'll admit, for the purposes of this demo, to get this running like faster than 20 minutes, you can run 10 populations and you do see a little bit of a plateau start to develop. But I just kind of want to get your all's opinion about what you think the number of ancestral populations we should use is, or if you can actually assess anything from this data.
Maybe it's. Yeah.
Cool. Anyone else? Any thoughts? Shake head, nod head. Yeah, so if you did include more data set, I think in this example, 5 actually with more data looks a little bit lower, with more K groups. But 6, I think is fair. I think saying that there are 6 ancestral populations makes sense based on what we see here. And so I actually switch based on what you guys have decided this matrix. So now we're going to retrieve data from our Q matrix, which is generated by TESS. And so really quick, let's just take a look at that. So this test3.object was generated by TESS, which is a large list. And so this, if we take a look at it, has our 1 through 7 population potential number of Ks that we've analyzed. And so each of these Ks, for example, I'll choose this, let's choose the 3. We have our number of populations, which is 3 ancestral populations. And then we have our cross entropy and our cross validation scores. And then we have the actual data that went into it. So that 170 individuals, 26,000 SNPs, ploidy of 1, ancestor groups, and then your Gs and your Qs that you generated based on that, that equation that I showed a little bit earlier. So test object has a lot of information, which I'll get into some of that in a couple of minutes. But first, we'll retrieve this Q matrix with those Q values for our ancestry K of 6. So we got that. And first thing we're going to do here is represent that data in a way that's really commonly used in structure with those bar plots. And so, oh, I have 5 colors. Choose a color. Someone choose a color. Purple. I already have. Oh, I guess purple works. All right, so we'll choose 6 colors, assign that to a palette, and then plot that with our individuals. All right, so this is our bar plot. It's very similar to what you would see in your structure types analyses, where you have your individuals grouped by their assigned ancestry. And so you have purple and the violet were really close together. But the ancestries are grouped together. So we have this purple group, this red group, this green group, this violet group, this blue group. Yes, and this yellow group. And so how many people here have seen something like this bar plot? Cool. Yeah, so pretty standard way to represent this. And then we have our structure-like pie charts that we can then use to plot our data back on the map. And then we'll loop through plotting a pie based on those ancestry coefficients. And so that looks something like this. So now we have our structure-like plots that show our assigned ancestry per individual to each of those 6 ancestral groups. I apologize for the number. I can actually get rid of that with the PyGlyph package from R. But again, these types of like pie charts that you see really often in things like your structure admixture types analyses. And then we can see some patterns start to emerge here where we have like some of these violet groups in one region, we have some of these red groups in another.
Do the coordinates have to be the same, or will it group like they're close-ish together?
Are you talking about just for the plotting of the actual pies?
Yeah.
It'll overlap. So like if they're too close, then you'll get pies that like are really overlapping like this, but you can adjust like you can adjust like the grouping or the size of your pies if they're like too big and overlapping with each other. All right. Cool. So that was our structure-like plotting. So this is again that like La Cava paper. So we have the same similar pie charts or bar plots in the pie charts. So now because we have that spatial component of our data, we can interpret like those values on our map as a gradient. And so what that ends up looking is something like this from that La Cava paper. So we can go back to R and then interpolate that map of ancestry coefficients. And then grab that. Cool. All right. So this is now with our 6 ancestral population clusters, what our assigned ancestries look like on a map. What's really cool about this is that if you're running tests and using it early on as some sort of preliminary visualization of your data, you can really easily start to develop hypotheses about what might be going on in terms of what's shaping your population structure for your across your study region. And so like even just looking at some of these purple or like violet, or this is purple, this is violet. I'm bad with colors. Purple was a bad choice. I apologize. I should have just shown something else.
Or maybe that's purple over there.
Yeah, it's purple.
And the blue.
So let's look at this red color. But we have, so you have some like geographic barriers or some sort of something happening geographically or for some reason in your population that is delineating this red cluster against these other colors. What's kind of cool about TESS is that you can then find individuals that might have been dispersed or like ended up in a completely different area than you would expect based on some sort of dispersal or other mechanism. And then find places where you have finer levels of structure that might be occurring in your population in some sort of geographic space. So I really like about this. I'm using TESS to help develop hypotheses for one of my dissertation chapters. And so what I've actually found is that when plotting this on a certain region of like my study site, is that these population clusters are basically defined by major interstates. So like those interstates like essentially shape what you see in terms of those population assignments, which I think is really, really cool. And yeah, so this is kind of like the big, like the important takeaway that you get from TESS is that you can visualize this and then use this for some of these, for generating those hypotheses about your data. All right, so we have 5 minutes left. Debating if I have enough time to go through a couple of like quick lines, or what do you think?
I mean,
Okay, cool.
If they're truly.
Yeah, they're quick. Yeah, so let's see. Yeah, so one example, so what we just did, actually, I think it's, Yeah, I'll play it by ear. But what we just did was assign those population, those ancestry coefficients, and then visualize that on a map using interpolation. And so this was done by Gustafsson et al. In 2018. So they had permaconcolor in California where they decided that 10 population or 10 ancestral clusters best represents their populations in California. And what they determined was that that genetic structure was highly influenced by urbanization. So for an example, if you look down here at San Bernardino, the Central Coast, and basically around the Los Angeles area, you have these smaller scale population clusters that are assigned compared to some of these more connected larger clusters. And so what they found is that the urbanization or the degree of urbanization in these areas really contributes to that population clustering that you see, which is what we see in other studies of permaconcolor in the southwest of California as well. So like I mentioned, that's really, really useful when generating those hypotheses, which you can then use to move on and try to assess, are there signatures of adaptation that might exist in those populations that are contributing to fitness more so in some individuals than others to things like urbanization or other environmental factors. What's really interesting is that in some of those populations, the genetic diversity was as low as what is seen for the Florida panther. And so that's really, really fragmented population when it gets down to those scales that were affected by that urbanization. So very, very applicable to these conservation types of studies. All right, 3 minutes, I could do this. So TESS is also really useful because it can identify outlier loci. So I think someone had mentioned that they were trying to find regions where you might see grouping of certain loci, especially loci that might be under selection or highly represented, or overrepresented in your data. And so what TESS does is it uses those ancestry coefficients to generate your FST values for each of your loci, and then generates p-values using by controlling for false discovery rate. And so I'm going to grab that little oh, there. Let's see. I just made this big. Yeah, so I'll go here again. We'll look at this, and then we'll look at our clusters. And so again, with our test run, we have all of our data, and that data includes our Q, our ancestry coefficients, and it also has our FST data represented by this FST as well. What's really useful about that is that you can then go in and then generate p-values for those FSTs based on the distribution of your FSTs in your data, and then see if there are any FST values that are outside of the range that you would expect based on your data. And so in this, we just have a histogram of p-values. We see that there's some p-values, or at least a good amount of p-values that have, or a good amount of individuals or loci that have p-values that are possibly contributing to something like adaptation. So we can then run our analysis of our p-values and control for false discovery rates. So we'll do that with these 5 lines. And then we can see the number of potential outlier loci that we find. So that's 473 there. And then finally, the last thing that we can do is plot this using a Manhattan plot. So here we'll plot all of our loci and their FST values, or their p-values that were generated based on their FST values on a plot here. And so we, again, have about 26,000 loci that we're looking at. And then we can then highlight the loci that might be under selection. And so now we can go in and then visualize across our data where areas that might be under linkage disequilibrium, or areas where SNPs might be under selection, and then use that moving forward in terms of that if you want to like scope down our study a little bit, like come back and say it's like, okay, it looks like there's something cool going on within SNP 0 to 10,000 or 1,200 or 12,000. Let's go in, like just subset that data and then try to see what these might be doing. But it's really, really cool way to do these quick preliminary assessments of these outliers. I think I have the last slide that I mentioned this. Yeah, so I do want to mention that when you do these types of outlier associations, that again, this might not be a process of adaptation. This could be a process of some of these things like population structure as well. I think that's one of the important takeaways here. So that being said, it's still useful when generating the hypotheses, which you can then use to test based on other data, like environmental data that you can generate. Tests in itself, what we saw here is that in the course of an hour, really 50 minutes, we were able to analyze 25,000 SNPs over 170 individuals, generate ancestry coefficients, plot those ancestry coefficients on a map to develop hypotheses, and then generate FST values and P values that we can then use to assess potential outlier loci and possible selection. So really, really useful tool. I'd recommend comparing it with other tools like structure and mixture if you can as well. But with that, I'll take any questions if anyone has any.
The
Yeah.
outliers run per population, right?
Outliers are run per loci.
Yeah, but does it?
Oh, the FST. Yeah, so
Does it take the population structure that you're discovering into account?
yes, so this is based on the 6 clusters that we selected, and it's assigning those individuals based on their cluster assignments. Yeah, other questions?
Basically right on time, so yeah, we'll live with that.
Cool, thank you all.
Yeah, thanks for joining us.